Question 2. Yeast cells can respire aerobically or anaerobically. A student used the apparatus shown in figure 3 to measure the rate of respiration in yeast. She positioned the flask in a water bath so that the yeast culture could reach a constant temperature, and then she left the apparatus for 1 hour before starting her investigation. Then we're given a diagram that will show us that the yeast culture is in glucose and there is capillary tubing with a coloured liquid. Question 2.1 asks us to suggest one reason why it is important that the student left the apparatus for 1 hour after the yeast culture reached a constant temperature. And this is so that the oxygen is used up, or the oxygen is then absorbed, or the oxygen is respired. For 2.2, it wants you to explain why the liquid moved to the right during the investigation. So that means that the coloured liquid has moved further along, which suggests that there has been a gas produced during the experiment that is forcing the liquid along the tube. So we can conclude that during anaerobic respiration, carbon dioxide is produced. It's anaerobic respiration because the oxygen that was in the flask has been used up earlier on and absorbed in that first hour when it was left. And we know that anaerobic respiration will produce carbon dioxide. This will then increase the pressure or the volume of the gas, which will then move the liquid further along the capillary tubing towards the right.